Hi, so this is a brief review of amino acids and disposal of nitrogen. So we'll be talking about degradation of the amino acids and then ultimately what happens to the nitrogen disposal. Okay, so to begin, amino acids, unlike carbohydrates and lipids, they are not stored. So there's no protein that exists that's just sole functions to maintain an amino acid supply for the body. So they got to be obtained from our diet. And if not obtained from the diet, we can also synthesize them on demand, or produce them from normal protein degradation. So in just recycling proteins, we can kind of reuse those amino acids. And then any excess of amino acids above and beyond the need for the body will get degraded and could ultimately be incorporated into triacylglycerols or ultimately turned into glucose for gluconeogenesis. And the nitrogen component to the amino acids, the ammonia, the NH3, is actually toxic in its free form. And so the liver will have a series of enzymatic reactions in order to take that ammonia group and combine it to form urea, which should be not toxic and can ultimately be excreted in the urine. So to begin, we'll talk about protein degradation. Now, most cells in the body are going to have what they call a ubiquitin proteasome proteolytic pathway. So these proteasomes in the cytosol can degrade proteins. Now, these proteins, you can see one which kind of looks like a blue scribble over here, and there's these ubiquitin molecules. And the ubiquitin, as it's aptly named, is really pretty much found everywhere in all these cells, and they can mark a protein for degradation by binding to it, which is an ATP-dependent process. This will essentially mark that for the proteasome to take that protein, unfold it, remove the ubiquitin so it can be reused again, and then basically bring it into the core of that proteasome and start to break it up into its amino acid components. Now, again, most cells will be able to do this, and so I'll be using the example of skeletal muscle, since that does have a lot of rapid protein turnover. So to start off, we'll talk about first off our ingestion of proteins. And so those proteins, which we refer to as dietary proteins, when they enter into the stomach, will begin to digest these proteins into sort of component parts. Ultimately, as we get into the intestines and the pancreas releases a series of enzymes, we can break them down to oligopeptides and amino acids, and then finally into the component amino acids that can be absorbed by the intestinal cells and then transported to the liver via the hepatic portal system. So on this slide, what I really want to start with is talking first about the pathway for a fed state. So I did mention from the previous slide, right, that we are here. Okay, let me get my pointer up. There it is. All right, so we're in a fed state there. Sorry about my handwriting again. And this is where, via the diet, we'll take in some dietary proteins. All right, and let's just draw my digestive tract here. All right, so this represents the gut. Okay, and so then there's ingestion of that. Ultimately, what's getting absorbed are the amino acids, which I'll label as AA over here. Okay. Now, I told you that they go into the hepatic portal system, so I'm going to draw a really large liver over here. All right. So here's the liver. And so these amino acids make their way to the hepatic portal system. All right, I'm just going to draw a line going through the liver first because these amino acids ultimately are going to pass through the liver, and many of them are going to be taken up by the hepatocytes. And others which will not be taken up by the liver can ultimately go to other cells in the body, which I'm just going to draw as a little box over here. We'll just say cells. These would just be generic cells of tissues somewhere else in the body. So now the amino acids that go through the liver and do get taken up, some of those amino acids will actually be converted into proteins that the liver will synthesize. And at the same time, some of those proteins could ultimately end up just being metabolized, ultimately to produce energy, and so we'll be producing a little bit of CO2 from that. However, if there's excess in the diet, all right, so this pathway is just going to represent some excess right here, excess proteins. All right, those excess proteins can be metabolized and converted into triacylglycerols, which we talked about that pathway before. Okay, the triacylglycerols can ultimately be packaged and secreted as VLDLs. So again, excess of anything, whether it's carbohydrates, lipids, or proteins, can all be converted ultimately into lipids, like triacylglycerols, and packaged and stored into the adipose tissue. So these VLDLs could be destined ultimately for adipose tissue storage. And at the same time, the excess, sort of that excess pathway, we could have glucose. We can make glucose from it, right? So we have gluconeogenesis in a fasting state, but since we're in the fed state, a lot of that glucose is actually going to be converted into glycogen. All right, and then the peripheral cells will produce some proteins that are necessary for other cells of the body. All right, so just to reiterate, we get the dietary protein, break it down to amino acids. The liver gets that first pass, all right, through the hepatic portal system, and then it can convert those into proteins for its own needs, break some of them down to energy, which I'll have labeled there as the CO2, and any excess could be ultimately converted into triacylglycerols, packaged into VLDLs, and sent off to the rest of the body. In addition, we can take some of those amino acids and actually convert them into glucose, and then in a fed state, we would take that glucose and put it into glycogen. Now, converting it into glycogen, again, the glycogen stores in the liver are limited, and ultimately, most of those, if there's an extreme excess, would probably end up as triacylglycerols in the VLDLs and shipped off to the periphery. Okay, so that's the fed state. All right, what's happening to proteins in the fed state. So what I want to talk about right now is what's going to happen here now in a fasting state. So we're going to say fasting. So we're in a fasting state now. And so I want to draw a muscle over here. All right, there's the belly of the muscle. All right. I'm going to draw a kidney over here. All right, there's the hilum, and you can see the ureter right there. Okay, so that's a kidney. I'm going to use the same kind of weird drawing as before for the gut. All right, so that's just going to represent the intestines. And then way over here, I'm going to draw my liver. All right, so that's the liver. So let's just make sure I label all of this so we know what is what. So that's the liver. There's the gut. And here is the kidney. And this would represent, let's just say, skeletal muscle. Okay. And so during a fasting state, I talked about this with you guys before, where we are going to break down some proteins in the muscle, and we're going to ship off some of those amino acids or secrete those amino acids into the bloodstream, which are ultimately going to make their way to the liver for further processing, where we can turn those into glucose through gluconeogenesis to keep our blood sugar levels high. But some of those amino acids could also go to the kidney and to the gut. So let's talk a little bit about this pathway. So in the muscle, we have an abundance of protein. And again, it's not the only source for proteins, but it's one of the classic examples because the muscles contain so much protein. These get converted into amino acids. Again, it could be using that ubiquitin proteasome pathway that we described from the earlier slides. And we break that down into the amino acid components. And now those amino acids could ultimately be recycled and reused by the muscle as well. But at the same time, we could actually take those amino acids and secrete them into the blood. So I'm going to draw here just a circle and put TCA for the tricarboxylic acid cycle, or the Krebs cycle. And from the Krebs cycle intermediates, we could ultimately form one of the main amino acids you will find in the blood, which is glutamine. All right, so glutamine is one of the main amino acids you'll see in the blood. They can also take the amino acid and convert most of those amino acids. They can convert it into alanine. And there will be some source of, we'll just put other amino acids. They're not all going to be converted into glutamine or alanine, but you will see that glutamine is probably the most abundant, and alanine following closely behind, in that they are really the way in which we're going to transport ammonia. All right, they're really going to be bound to ammonia, and these other tissues are going to utilize that. So, for example, glutamine, which, as I said, is very abundant, can go to the kidney. There you go. So it goes to the kidney. And I've actually discussed this pathway with you guys already, but glutamine can be taken up by the nephron cells, or the cells lining the nephron, excuse me. That glutamine can be cleaved and release. All right, it can release NH3. NH3 can then become NH4 in the lumen of the nephron when it binds to a proton. So it actually acts as a buffer for the protons that get secreted, because if you recall, the protons can't really stay as free protons in the urine, so it gets bound by this ammonia buffer. And then now the glutamine is no longer glutamine, but can be converted and become alanine, just like we see over here, the alanine. The glutamine as well can also go to the gut, which at that point the glutamine can be used for fuel. And oftentimes it does get converted to alanine as well. Alanine. All right, so it can be converted to alanine as well. And so what we're having is processing through the kidney, through the guts, of taking glutamine, ultimately converting it to alanine. And the alanine has also been released directly from the muscle, all making its way to the liver. All right, it's all kind of going to converge over here. So let me just draw that as converging. All right, so we converge. We can converge over here. And this can ultimately be processed. And we can remove the ammonia group and form urea. So now the liver is the spot where urea will be processed or be formed. Okay, so we don't normally transport ammonia as free ammonia in the bloodstream, because the ammonia could actually tamper with our homeostasis, because it can affect the pH of the blood, since it acts like a buffer. So we typically are going to transport it as an amino acid, and that amino acid would be glutamine or alanine typically. Not the only ones, but typically. And so the glutamine and glutamine can go directly to the liver as well, mind you. All right, so whether it's alanine or glutamine making it to the liver, ultimately we can cleave off some of that ammonia and produce urea from that. And we'll talk about the urea cycle later. In addition, as you guys know, during a fasting state, I can also make glucose. So gluconeogenesis can take place. I can also make ketone bodies from that and release that to various tissues. So you know the ketone bodies can be used by muscles, kidneys, and ultimately the brain if the fasting is very prolonged as well. Okay, so what I want to get to now is talking about the removal of the nitrogen from the amino acids. So again, we're talking about that degradation process. So something you would see like during fasting, like we talked about on the previous slide. So we're going to take the ammonia that essentially we want to turn into urea, transport it as glutamine or alanine primarily, but there are other ways to transport as well, but we're not going to get into detail on that in this particular lecture. So we'll transport that to the liver, where we can ultimately produce the urea. So there's an important process that has to take place. So the important process here is called transamination. So this is ultimately where we're going to take most of the amino acids, all the different kinds that there are, and we're going to convert them into essentially glutamate. All right, so it's like we call it like a funneling process. And so this is how we're essentially going to take any amino group, all right, convert it to glutamate. And the way we're going to do that is using these aminotransferase enzymes, where there'll be specific ones for a given amino acid. So if you look down here in the box, the cartoon, the alpha amino acid, so that's whatever amino acid we're talking about, all right, can interact with an intermediate like alpha ketoglutarate. which you may recognize, that's actually an intermediate in the Krebs cycle. So if it transfers an amino group to alpha-ketoglutarate, that produces glutamate. And the amino acid that gave off the ammonia actually becomes some sort of alpha-keto acid, which is an intermediate it could be an intermediate in the Krebs cycle or part of glycolysis or some other type of intermediate. And these enzymes are referred to as aminotransferases, and there'll be specific ones for amino acids. And it also requires this cofactor called PLP, or pyridoxal phosphate. And the pyridoxal phosphate is a vitamin B6. You'll notice that the arrows here actually face both directions. So this actually can be going either direction, where we can take glutamate and convert multiple different types of glutamate into different amino acids, depending on what glutamate interacts with, what alpha-keto acid interacts with. So not only is this pathway used for sort of funneling and breaking down amino acids and converting them into glutamate, but it can also be used to synthesize new amino acids. So it's an important process. This is what we refer to as transamination. So what I want to draw your attention to right now, let me get my pen ready here. All right, I want to talk about the Krebs cycle real quickly. So if you guys recall, in one of the lectures I talked about oxaloacetic acid, which combines with acetyl-CoA. All right, and so we get a combination of these two, which forms citrate, and then we form isocitrate, which can then become alpha-ketoglutarate. And alpha-ketoglutarate is this guy right here. So he, all right, is an intermediate in the Krebs cycle. And there'll be other intermediates that we can come back to, but ultimately, you know that we have several other reactions going on here and converting us back into oxaloacetic acid, or OAA as I have it here. Okay, but I want you to recognize that alpha-ketoglutarate is actually an intermediate in the Krebs cycle. All right, so again, when an amino acid, some other type of amino acid, all right, interacts with alpha-ketoglutarate, it's going to produce some sort of intermediate alpha-keto acid. Those intermediates vary depending on what the amino acid was to begin with. And the alpha-ketoglutarate from this aminotransferase reaction then becomes glutamate. So glutamate ultimately picked up, or excuse me, alpha-ketoglutarate picked up the NH3 molecule. So we transferred it from one amino acid to another and converted it into an intermediate. Okay, so there are multiple different reactions that can occur for the various amino acids and converting them into glutamate. So there's different aminotransferases. What I want to do is demonstrate two very important ones that you guys should remember. So, let me get my pen ready here. Okay. All right, so this process, okay, again, is referred to as transamination, because we're basically handing off an ammonia group here. All right. So again, if I take any amino acid, I'm going to give you very specific amino acids because these particular amino acids are important ones to remember. So the first one is alanine, and you'll recognize that because I talked about alanine earlier on as being one of the amino acids that transports ammonia through the blood. So let's take alanine for example, and if it interacts with alpha-ketoglutarate, all right, from our Krebs cycle, all right, in the presence of the aminotransferase, all right, what's going to happen is the alpha-ketoglutarate, as you already know, is going to become glutamate. All right, that's our primary amino acid there. That's going to, we now have an NH3 group connected to it. All right, and the alanine, well, the alanine, that what it gets converted into after it loses its ammonia group is actually pyruvate. And as you guys know, pyruvate is the end product of glycolysis. All right, so that actually makes alanine a very important step here. So pyruvate's an important intermediate. All right, so remember that comes from the end product of glycolysis. So that's what it gets converted into after transferring its ammonia group. And I really should draw 2 arrows here because these reactions can go both ways. Now the aminotransferase, all right, is actually very specific. For this one, let's call it an alanine aminotransferase, or ALT. And of course, it will require PLP, which is that cofactor, the vitamin B6. And you guys may recognize aminotransferase ALT. All right, I'm going to come back to that. All right, so the next one I want to talk about, the next amino acid is aspartate. All right, so aspartate can interact with alpha ketoglutarate, and you already know the reaction. Alpha ketoglutarate will become glutamate. Glutamate. And aspartate, in the meantime, all right, that will be converted after it passes off its ammonia group. It will become oxaloacetic acid. And as you guys may recognize, oxaloacetic acid is what combines with acetyl-CoA at the beginning of the Krebs cycle. So we recognize that intermediate as well. And aspartate's another important amino acid, which I'm going to come back to because it's important for the urea cycle itself. All right, and its aminotransferase, aspartate transferase, or AST. All right, and it also requires PLP, which is the vitamin B6. And you'll notice here the ALT and AST. Those of you who are more clinically familiar, all right, may recognize that sometimes we take AST and ALT levels when we're looking at liver function. So these are actually aminotransferases because these enzymes are found in abundance in the liver, and also you can see them in other tissues as well. But they are a good marker of liver function, and so when they're usually increased in levels in the blood when there's liver damage. So there's good correlation with the AST and ALT. So that's actually what those, what the property of those AST and ALT are. All right. So here on this slide, you see that the 2 important aminotransferase reactions that I just discussed here, ALT, all right, present in many tissues, reversible, all right. AST is also reversible, comes from the aspartate. It's used as a source of nitrogen in the urea cycle, which I'm going to come back to. And again, we use these levels clinically to help us assess the health of the liver. So the question is, after we've funneled these various amino acids through aminotransferase reactions and convert them into glutamate, what do we do with glutamate then? So here, glutamate will have a specific enzyme. All right, so let me get my pen ready here. So we form glutamate, all right? And so in particular in the liver, and you can also see this in the kidney, we can take glutamate and we may convert it back now to alpha ketoglutarate, so back to its intermediate form. All right, but what we're going to do in the process of reforming alpha ketoglutarate is we're going to free an ammonia group from the glutamate, right? Because once I remove the ammonia from glutamate, it becomes alpha ketoglutarate. So in order to do that, it'll require NAD. So that's going to become NADH. Okay. And the enzyme responsible for this is glutamate dehydrogenase. All right. So glutamate dehydrogenase. So that's an important enzyme for you guys to remember. So these cells, like the liver, for example, the hepatocytes, they're going to have glutamate dehydrogenase. So they can take glutamate, which has this ammonia group, and cleave off the ammonia, and now we're left with a free ammonia group. All right, so I'm going to put a little quotations here. It is free ammonia group that ultimately can be used in the urea cycle. So we're going to take that ammonia and turn it into urea. Now, the liver is not the only one that has glutamate dehydrogenase. Other cells will have it as well. And I'm going to show you some cycles that can occur in those cells to ultimately produce free ammonia that will be used to produce a different amino acid. So we'll talk about that next. But first, this is just showing you the cartoon. the glutamate dehydrogenase. You can see there's the reaction glutamate converting into alpha ketoglutarate, which requires our enzyme, glutamate dehydrogenase, which frees up an ammonia group right here, requires our NAD and NADH. The reaction is reversible. You could take alpha ketoglutarate and convert it back into glutamates, which would just be adding the amino group back, but requires the reducing agent NADPH right here. Okay. Okay, so I want to talk about the fasting state. So here, you have a pen ready. All right, so we're going to talk about fasting. All right. Now, if you can recall, during fasting, we're breaking down some of those proteins, and we're going to move the amino acids into the bloodstream so that ultimately in the liver we can produce glucose from them. And then as a side product, a byproduct, excuse me, of that gluconeogenesis and breaking down the amino acids, we're going to be producing ammonia, which is going to be converted into urea and ultimately excreted in the urine. So during the fasting state, what I want to be able to do in the skeletal muscle is when I break down the protein into those amino acids, is I want to transfer that ammonia to the liver, right? And most of that's going to be bound as either glutamine or alanine. So if you recall that from our earlier slides. All right, so I want to show you some of the steps in which I can take my glutamate. All right, so here's glutamate. And so glutamate, I can actually convert to glutamine, which would be one of the ways in which I'm going to transport ammonia. And what I'm actually going to do is I'm going to add, that's going to require, excuse me, adding an ammonia group to it. So I'm going to need a free ammonia group, add it to glutamate, and it'll become glutamine. So we'll have two ammonia groups, all right? And the enzyme is glutamine synthetase. All right, so glutamine synthetase is going to convert glutamate into glutamine. Glutamine now has two ammonia groups, and it's going to be released into the bloodstream, where it'll ultimately end up in various tissues like the kidney, the intestines, and even the liver, right? So that's one. The other one is I'm going to take glutamate, and if I want to transport it as alanine, I kind of already showed you this aminotransferase process. I can convert glutamate, excuse me, back into alpha ketoglutarate. All right. And I can take glutamate when it passes off an ammonia group to pyruvate. Pyruvate's going to give me, okay, alanine. So now I'm forming alanine here. All right. When I formed alanine and I formed glutamine, and I can do that in the skeletal muscle. I can do that in the skeletal muscle, and I can release glutamine and alanine. And then that can be transported to the liver. And so these will be sort of our primary amino acids, not the only ones, but the primary ones. And so they'll go to the liver, and in the liver, ultimately, all right, it's going to end up in is either glucose for gluconeogenesis, plus we're going to create some urea from that. Okay, and so this is going to happen in the liver. Okay, so the ammonia transport to the liver is again mostly going to be through this glutamine or alanine. So I have the two mechanisms there: glutamine synthetase, so we're going to convert glutamate into glutamine by combining it with another ammonia group, and then the formation of alanine via the transamination from pyruvate. Okay, and this cartoon on the side here I like. We're really going to take a look at two different cycles. So we're going to use skeletal muscle as our sort of example here in the peripheral tissue, and then here's the liver. Okay, and I have two groups here I want to talk about. So there's two different cycles. So you're going to have one where in the skeletal muscle we're going to release glutamine, which is this GLN down here. Glutamine, it's going to be released to the bloodstream, and then the glutamine is going to become or reconverted back into glutamate. All right, and we're going to cleave off that ammonia group in the liver. All right. So we'll talk about how that's done in just a minute. and then that ammonia is going to be used to produce urea, and the glutamate can actually be re-released back towards the skeletal muscle and re-enter the cycle to produce more glutamine. So we get a glutamate-glutamine cycle between the muscle and the liver. Similar to that, we can take other amino acids, right, and we can convert them into alanine instead, and then the alanine can be shuttled to the liver, and then the liver can convert that into pyruvate. The pyruvate can be turned into glucose via gluconeogenesis, and then the glucose can be sent back to the muscle, and the muscle can reconvert that into alanine. And so you have a glucose-alanine cycle. And so this is a way we can kind of keep this particular degradation pathway going. So let me describe that or draw it out for you on the next slide. Okay, so let's first talk about the glutamine-glutamate pathway. So again, this is going to be breaking down amino acids in the skeletal muscle, all right, converting them into glutamate, ultimately to glutamine. Glutamine can then be transferred to the liver, and then the liver will cleave off that ammonia group, converting it back into glutamate, and the glutamate will be sent back to the skeletal muscle to be reconverted into glutamine. And so we kind of have this shuttling process that's occurring. So let's describe that sort of a piece at a time here. All right. So if we're looking at this pathway, we can take any amino acid, all right? Now, not all of them will go through this process, but this is one of the main ways of doing it, so we're going to focus on this. So that amino acid can ultimately be converted into some sort of alpha keto acid or intermediate, okay? Some of them we described, like alanine and aspartate, for example. And then if we combine that with the alpha ketoglutarate from the Krebs cycle, all right, we can make glutamate. We can make glutamate. Once we make the glutamate, now remember glutamate's going to be carrying that ammonia group now. So we've got transfers to the alpha ketoglutarate to make glutamate. We're going to draw another cycle here. We're going to convert glutamate back into alpha ketoglutarate, and we're going to remove the NH group that it just picked up. So we're going to remove that ammonia group right away. So some of that's going to, the glutamate's going to be converted back into alpha ketoglutarate. We now have free ammonia. Right, so we have free ammonia. That was a dehydrogenase reaction, so I'm going to put a little water symbol there. Okay. And here we're going to now take that free ammonia group, and we are going to combine that ammonia group, all right, to another molecule of glutamate. So a proportion of the glutamate was created via other amino acids from alpha ketoglutarate, and then we took a proportion of that glutamate that was produced, and we converted it back into alpha ketoglutarate by freeing up an ammonia group. And we'll take another portion of the glutamate that's there, and we'll actually give it another ammonia group. All right, so we'll give it another ammonia group, and we'll convert that into glutamine. And if you guys recall, this is glutamine, all right, synthetase. Synthetase, which is the enzyme that converts glutamate using a free ammonia to make glutamine. So that's how we're producing now glutamine inside the muscle here, inside our skeletal muscle. So let me put that over here to make sure we remember where we are. Skeletal muscle. Okay, so we just produced glutamine, and again to free up this ammonia from a glutamate that was glutamate dehydrogenase, okay, and then over here we had an aminotransferase enzyme plus PLP. So it took an amino acid, okay, removed its amino group, added to alpha ketoglutarate, produced glutamate. We recycled that. We took the ammonia group off of some of that glutamate, reproduced alpha ketoglutarate, took the free ammonia, combined it with another portion of glutamate with the enzyme glutamine synthetase, and produced glutamine. Okay. Now the glutamine can then go into the blood. All right, so it can go into the bloodstream. All right, go to various tissues, be used by something like the kidney or the intestines. But in this example, it's going to go to the liver. All right, so we're here in the liver down here, and we're going to take that glutamine. So we take glutamine, and we're going to reconvert it into glutamate. If you remember, the way we're going to do that, or I'm sorry, I say remember, but I actually have not talked about this enzyme yet. All right, we're going to need to we're going to be removing an ammonia group. All right, and we're going to need an enzyme referred to as glutaminase. Glutaminase. So this glutaminase enzyme removes an amino group from glutamine, converting it back into glutamate. All right, and I'm going to now use water over here. All right, so now we've reconverted it back to glutamate, and this free ammonia over here in the liver will be used for urea. All right, over here back in the muscle, the free ammonia was used to combine to glutamate using glutamine synthetase to make glutamine, then transporting that free ammonia. So technically, what we did was we transporting free ammonia that was produced in skeletal muscle. All right, skeletal muscle cannot produce urea, so it takes that free ammonia, combines it with glutamate to make glutamine, which gets transported to the liver. The glutamine then is cleaved to remove that free ammonia group, which can then be used to make urea. Now we've reproduced glutamate, and then glutamate can actually make its way back, all right, into the bloodstream, back to the skeletal muscle, therefore completing our cycle. So that glutamate can be reused to pick up another ammonia group and glutamine. In the meantime, the amino acid that gave off that ammonia, the initial ammonia group from the beginning of this whole cycle, all right, over here, all right, will have turned into an alpha keto acid, which can be converted or processed and put into the Krebs cycle there and been used for energy. Or in some cases, even the glutamate that we produce here may not go back to the skeletal muscle. Instead, we can actually break that down or turn it into alanine or make it, an intermediate part of the Krebs cycle for either energy, but most likely in a fasting state to turn that into glucose. So that covers the glutamine-glutamate cycle. The last cycle I want to talk with you guys about is the alanine, glucose-alanine cycle. So we're going to start in skeletal muscle again. So this is skeletal muscle. We're going to talk about this cycle one more time. So we're going to take an amino acid, convert it into its alpha keto acid intermediate. As you know, we're going to start with alpha ketoglutarate. Alpha ketoglutarate is going to give us glutamate. So glutamate picked up that ammonia group. All right. Now in this process, we are going to convert glutamate back into alpha ketoglutarate. All right. In exchange, glutamate is going to give off its ammonia group to pyruvate. Now this is actually an important step in skeletal muscle because skeletal muscle tends to have a lot of energy and requires a lot of energy, excuse me. So it usually has a lot of pyruvate intermediates, so from glycolysis. So since skeletal muscle undergoes a lot of glycolysis, there's usually a ready amount of pyruvate. So the glutamate can pass off an ammonia group to pyruvate. And the pyruvate then, of course, having attained that ammonia will become alanine. All right. Now the alanine can then get transferred in the blood to our liver. All right, so we're in the liver now. We're going to take that alanine and we're going to reconvert it back into pyruvate by passing the ammonia group to the alpha ketoglutarate in the liver to make glutamate. All right, and this pyruvate can ultimately be turned back into glucose, and then recycled back in this direction. All right, back in this direction. So we get again a glucose-alanine cycle. So the glucose can come back and become pyruvate. So that glucose can be broken down by skeletal muscle into pyruvate, ultimately back into alanine, and so on. So that's the cycle that's occurring there. Meanwhile, the glutamate that we produced inside the liver over here, we can actually convert that back into alpha-ketoglutarate, all right, by removing an NH3. So free off the NH3. Again, we're going to need some water. This is going to be H2O, glutamate, dehydrogenase reaction that we talked about. Dehydrogenase. And now we have free ammonia that can enter the urea cycle. All right. So in both cases, what we're seeing is a cycle between skeletal muscle and the liver using glutamine and alanine, and ultimately being able to free up an ammonia group. So the alanine and glutamine kind of, in a sense, act like transporters of ammonia that can be reconverted and sent back, and the cycle can be continued. And so it drops off the ammonia to the liver, where we can then use that ammonia to produce urea. Now, in some cases, some of that will be part of the cycling process between skeletal muscle and liver. Other times, we'll take the alanine or take the glutamine and break it down further and use it for gluconeogenesis or energy or something like that. So in this cartoon here, you can see both those pathways. So what we have over here, up here is the glutamate being converted into glutamine. All right. So again, using glutamine synthetase. The glutamine sent to the liver. Using glutaminase, we remove the ammonia group over here, which is ultimately going to become urea. And the glutamine then is converted to glutamate. Glutamate dehydrogenase can also remove another free ammonia, which can go to urea, right? Which is going to produce more alpha-ketoglutarate. The alanine process, so over here you can see alanine, which is coming from the pyruvate. And so again, glucose being broken down into pyruvate can be processed into alanine by using alpha-ketoglutarate, the glutamate aminotransferase pathway. Alanine gets shuttled to the liver. Alanine can then be converted back into pyruvate, converted back into glucose. So that's our shuttle there. All right. And what this also is showing in this cartoon is that there are other amino acids that can enter in and become part of this glutamate alpha-ketoglutarate pathway, these aminotransferase pathways. So there are some exceptions to every rule, and you'll see there's some isoleucine and valine, which can enter in and form succinyl-CoA as opposed to interacting directly with the glutamate, or excuse me, the alpha-ketoglutarate. So there are, alternative routes or intermediates that can be produced from the breakdown of some of these amino acids that can be converted into other intermediates of this pathway. So this is just showing you here glutamine. So converting glutamine into, so here's our glutamine, converting it into glutamate. All right, using glutaminase, which I demonstrated that on the drawing for the liver. All right, this way we can free up an amino group, and that can be part of the urea cycle. And then here's glutamates. We can do the reverse, glutamate back into glutamine using glutamine synthetase. So that's something we saw, what I drew in the skeletal muscle, so that I could add an ammonia group to glutamate and transfer that ammonia to the liver. All right, so glutamine, the glutamine amide provides non-toxic storage and transport of ammonia, right? Because like I said earlier on, I can't transport ammonia directly as it would act like a buffer and affect homeostasis of the blood. The synthetase is mainly in skeletal muscle, liver, and also in the brain. So we've seen a lot of it in the CNS. It's a major way to remove ammonia, and it's a vital mechanism in the brain in particular. And glutamine is found in abundance in the plasma, probably at the highest concentration than any other amino acids. Alanine would be another one that's at higher concentrations as well. So now I want to talk briefly about the urea cycle. So what we described up to now is really, say, in a fasting state, taking different types of amino acids, converting them into glutamate, and then combining glutamate with an ammonia to make glutamine, which can then be sent to the liver, and we can remove that ammonia group and send it to the urea cycle. Or during the fasting state, we can convert any amino acid into glutamate, convert it into alanine, then alanine can be sent to the liver. And in the liver, it can then take the alanine, convert it back into pyruvate, make glucose again through gluconeogenesis, and send it back to the muscle. Meanwhile, that glutamate that we just made can then be cleaved using the glutamate dehydrogenase, remove the free ammonia, and then that can be sent to the urea cycle. So now let's talk about what's going to happen with those free ammonias. Okay, so now what I want to draw out for you is the urea cycle. So, pen ready here. Okay, so we have the urea cycle. Okay. So first off, I'm just going to draw here. This is going to be the mitochondria. All right, and what we're going to have is our free ammonia. So that's the free ammonia that we got again. That would have been cleaved from the glutamine as well as the breakdown of the glutamate back into alpha ketoglutarate. So the ammonia combines with bicarb, plus we're going to need some ATP. So this is how it starts. So we need the bicarb. So in fact, to make urea, we're actually going to be needing this carbon here from bicarb. And that carbon is going to be the carbon found in urea. All right, and we're going to use this nitrogen from the free ammonia as part of the urea complex as well. So the first step here, all right, is we're going to form carbamoyl, moyl phosphate. So carbamoyl phosphate, and that takes that enzyme carbamoyl phosphate synthetase 1, CPSI, all right, or CPS1. It's going to produce carbamoyl phosphate. Now carbamoyl phosphate, all right, is then going to combine with another type of amino acid referred to as ornithine. So it can combine with ornithine, and that's going to produce citrulline. All right, that's going to produce citrulline. Now citrulline is going to get transported out of the mitochondria. All right, citrulline is actually going to combine with, in the cytoplasm, the amino acid aspartate. And if you guys recall, I told you guys that from the first couple of slides that aspartate is going to be an important amino acid for the urea cycle. So the aspartate combines with citrulline to produce arginosuccinate. All right, arginosuccinate is going to be broken down into fumarate. Fumarate and arginine, which is another type of amino acid. And arginine is going to produce ornithine. So the cleavage of arginine is going to be, again, the cleavage of arginine is going to be via an enzyme called arginase. So that's the enzyme. It's going to produce ornithine and the final product, which is urea. So here's our final product, urea. So that required arginase. Now you notice ornithine was used over here as well. So ornithine actually gets recycled. So I'm going to point that out here. All right. So ornithine gets recycled all the way back. It's reused over here. Sorry, a little mess up there. So the ornithine gets recycled, binds to more carbamoyl phosphate, produces citrulline, and we re-enter this. And so now the ornithine combining with carbamoyl phosphate producing citrulline. The citrulline combines with more aspartate. All right. Now, another critical step here is the production of this fumarate, which is actually an intermediate in the Krebs cycle as well. So fumarate can ultimately become more oxaloacetic acid. And if you guys recall, the oxaloacetic acid produces aspartate. So if it combines with glutamate, so the glutamate, the ammonia group from glutamate, if it's transferred to oxaloacetic acid, glutamate then becomes alpha ketoglutarate, and we get aspartate. So we have this cycle going on where we can recycle ornithine, and the ornithine can then bind with more carbonyl phosphate. So we have a final product of urea. So let me just show you what the structure of urea looks like down here. So here's nitrogen, down to a carbon, to another nitrogen. There's a double bond here to oxygen. And now you don't have to memorize the structure, but I just want to point it out simply because I want to show you where we're getting each of these nitrogens and carbons from. All right. So here's the structure of urea, where you have a single carbon. That single carbon came from the initial bicarb from the free ammonia combination. So that's carbon from the bicarb. All right. And we have these two nitrogen groups. So these two ammonia groups, the first ammonia group came from the free ammonia. The second ammonia group came from the aspartate. All right, so the aspartate and free ammonia deliver ammonia ultimately to the urea molecule. And the urea gets sent into the bloodstream to the kidney to ultimately be excreted in the urine. All right, so it's a process that will continue to produce the urea as long as the amino acid cycle continues between the breakdown of proteins from the peripheral tissue being sent back to the liver. And as you know, if we were to extend the time period of fasting to several days, that this production would actually go down somewhat, simply because the ketone bodies levels would increase due to the fact that skeletal muscle stops using as much ketone bodies, and therefore the nervous tissue can use more ketone bodies and requires less glucose. So we don't need as much amino acids or as many amino acids for gluconeogenesis. And so this slide here is just showing you essentially the same pathway, but it's giving you the structures. So you guys can take a look at that in your notes here, but you can see the intermediate, the carbonyl phosphate, which is coming from, again, this green C from the bicarb, the carbon from the bicarb, the free ammonia, and some ATP. Then you have ornithine making for citrulline, and then citrulline combines with our aspartate over here. All right, and then the aspartate makes plus the citrulline produces arginosuccinate. All right, we'll produce arginine with arginase over here, which is going to produce our urea, and we'll recycle the ornithine. The fumarate over here, which was produced, all right, can ultimately become malate to become oxaloacetate, all right, or OAA, all right, which again through an aminotransferase with glutamate can become aspartate, and so we have this nice cycle going on of recycling the different intermediates. So again, the first two reactions occur in the mitochondria, and then after we produce the citrulline, that's going to occur in the cytosol. All right, so one nitrogen of urea is supplied by the free ammonia, the other from aspartate. And then arginase, which I mentioned already, hydrolyzes arginine to ornithine and urea. So virtually all this is occurring exclusively in the liver. All right. And then on a final note, just a clinical note, if you have liver disease or liver malfunction, that can result in impairment of production of urea and the ability to essentially excrete ammonia safely in the form of urea. So therefore, you can get increased ammonia levels. And ammonia can actually be directly neurotoxic. So somebody with a severe liver disease could show signs of severe central nervous system disorders like tremors and slurring of their speech, somnolence, vomiting, cerebral edema, blurring of vision, and so on, which can also, kind of look like things like strokes and so on. So at high enough concentrations, ultimately that could be deadly. All right. So if you guys have any questions, please email me.